Part 2 of mitosis, meiosis, reproduction, and early prenatal development. If we look at sexual development, during the 5th week of prenatal development, all embryos develop two sets of unspecified indifferent gonads, reproductive ducts, in the female Müllerian and in the male Wolffian. An embryo develops as a male or female based on the sex chromosome constitution and actions of certain genes, specifically the SRY gene, sex-determining region of the Y chromosome. The sex chromosomes determine our gender. Human males are the heterogametic sex with different sex chromosomes, XY. Human females are the homogametic sex, XX. In other species, sex can be determined in many ways. For example, in birds and snakes, males are homogametic, ZZ, while females are heterogametic, ZW. If we take a look at this next slide, you can see the paired duct systems in the early embryo may develop into a male or female reproductive organs. The red tubes represent female structures and the blue tubes represent male structures. Here we have a diagram of the male and female with the XY and the XY chromosome, what it looks like, and in the female, XX. And you can see what the X chromosome looks like and what the Y chromosome looks like. In looking at the X and Y chromosome, the X chromosome contains 1,500 genes. It's larger than the Y chromosome, as you just saw in that picture. It acts as a homologue to the Y in males. And the Y chromosome contains 231 genes. Many DNA segments are palindromes and may destabilize DNA replication. If we look at this picture of the anatomy of the Y chromosome, we see that the Y chromosome has two pseudo-autosomal regions, PAR1 and PAR2, and a large central area, MSY, that comprises about 95% of the chromosome, and it has only 22 genes. SRY determines the male sex, and AZF, which you can also see there, encodes a protein essential to producing sperm. Mutations in it cause infertility. So if we take a look at the SRY gene, we see that it encodes a transcription factor, which is a protein. This controls the expression of other genes. It stimulates male development. It sends signals to the indifferent gonads to destroy female structures. It stimulates the development of male structures. And testosterone and dihydrotestosterone DHT are secreted, and they stimulate the male structures. Here we see a diagram that is showing you the mutations that affect the male development. And in a normal male, in prenatal development, there's the activation of a set of genes beginning with the SRY, which sends signals that destroy the female rudiments while activating the biochemical pathway that produces testosterone and dihydrotestosterone, which promotes the development of male structures. The green arrows indicate where mutations disrupt normal sexual development. And there are specific diseases which have been identified here, such as 5-alpha reductase deficiency, congenital adrenal hyperplasia, and androgen insensitivity syndrome. In looking at the abnormalities in sexual development, androgen insensitivity syndrome is a lack of androgen receptors. Pseudohermaphroditism is the presence of male and female structures at different stages of life. 5-alpha reductase deficiency is an absence of DHT, and congenital adrenal hyperplasia is where there are high levels of androgens. This is a table which is showing sexual identity. In here you can see XY is a male, XX is a female, and the timing for this level is fertilization. Then we have gonadosex, which is undifferentiated structure begins to develop as a testis or an ovary. This is 6 weeks after fertilization. We have phenotypic sex. This is the development of external and internal reproductive structures. It can to continue as male or female in response to hormones. This occurs 8 weeks after fertilization and in puberty. And gender identity, strong feelings of being male or female develop from childhood, possibly earlier, and sexual orientation, attraction to same or opposite sex from childhood, it is. Y-linked traits are genes on the Y chromosome that are said to be Y-linked. Y-linked traits are very rare. They are transmitted from male to male. A female does not have a Y chromosome. Currently identified Y-linked traits involved infertility, and these are not transmitted. If we look at X-linked traits, these are patterns of expression on the X chromosome. They differ in females and males. Females have X-linked traits are passed like autosomal traits. In males, a single copy of an X-linked allele causes expression of the trait or illness. The human male is considered to be hemizygous for X-linked traits. Here we see a diagram showing you the sex determination in the females and males. X inactivation. Females have 2 alleles for the X chromosome genes, but males have only 1. In mammals, X inactivation balances this inequality in the expression of genes on the X chromosome. A female mammal is a mosaic for expression of most genes on the X chromosome. The XIST gene encodes an RNA that binds to and inactivates the X chromosome. X inactivation occurs early in prenatal development. It alters the phenotype and not the genotype. It is an example of an epigenetic change, an inherited change that does not alter the DNA base sequence. Here we have a diagram showing X inactivation where female is a mosaic for expression of genes in the X chromosome because of the random inactivation of either the maternal or paternal X in each cell early in prenatal development. In X inactivation, it's used to check the sex of an individual. The nucleus of the cell of a female during interphase contains a Barb body. A female is XX. She therefore would have 1 Barb body. The consequence of X inactivation on the phenotype is homozygous X-linked genotypes, no effect. In heterozygotes, it leads to expression of 1 allele or the other. In X inactivation in humans, it can be used to identify carriers of some X-linked disorders. We will be looking at this at a later time and talking about such diseases as Lesch-Nyhan syndrome, and it also affects the severity in Rett syndrome. Unequal X inactivation pattern can occur if the 2 X chromosomes have different alleles for a gene that controls cell division. This gives certain cells a survival advantage. If we look at the phenomenon of parent of origin effects, the parental origin influences the phenotype. It influences the age of onset and the symptom severity. Mechanisms of parent of origin effects are genomic imprinting and differences between developmental timetables of sperm and oocyte. In genomic imprinting, if you remember, we have the methyl groups, and the methyl groups bind a gene or several linked genes. They prevent them from being assessed to synthesize a protein. The parental effect on gene expression is seen as diseases inherited from a parent. Epigenetic alteration is where a layer of meaning stamped upon a gene without changing its DNA sequence. In looking at this diagram of genomic imprinting, we find that the imprints are erased during meiosis. Then they are reinstituted according to the sex of the individual. The importance of genomic imprinting is the function of imprinting isn't that well understood, but it may be a way to regulate the abundance of key proteins in the embryo. Imprinted genes are in clusters along a chromosome controlled by imprinting centers. One gene in a cluster could be essential for early development. Others become imprinted due to what is referred to as the bystander effect. Again, we see that research suggests that it takes two opposite sex parents to produce a healthy embryo. The male genome controls the placenta development. The female genome controls the embryo development. Genomic imprinting may also explain incomplete penetrance, which we will be discussing in a couple weeks. It also may be a concern in assisted reproductive technologies.